All right, guys, so today we're going to move on to unit number 2, which builds upon unit 1 that we discussed with mitosis and meiosis. And this is entitled Mendelian Genetics. So, what is Mendelian Genetics? Well, we're going to discuss that, but first, I'd like to show you our agenda for this presentation. So, we're going to talk about genetics and heredity. We're going to discuss Gregor Mendel. We're going to talk about some of the terms that you're going to need to define, which I've already posted on Google Classroom for you to go ahead and get started with. We're going to talk about monohybrid crosses, the patterns of inheritance, dihybrid crosses, what is a test cross, and beyond Mendelian genetics, incomplete dominance. So you might not know what those things mean, but we're going to talk about them in the coming days. So let's get started. Introduction to genetics. What is genetics? Genetics is a branch of biology that deals with heredity and variation of organisms. So in our previous unit, we discussed chromosomes and what those are, and they carry the heretic information. They're genes found within chromosomes, and they're what code for our specific traits and attributes that make us who we are. So let's talk about what chromosomes are on a little bit of a deeper level, and we're going to get into this in more detail in coming chapters, so don't worry if you don't get it all. So chromosomes have genes, which are what you see here, these different colored sections on the chromosome. And these genes are part of the DNA because we know that chromosomes are made up of highly tightly coiled DNA. And so genes are an arrangement of nucleotides, which are the individual building blocks of DNA. And DNA codes for RNA, and RNA then becomes the template to create proteins. And proteins are the heavy lifters in the body. They're the ones that do all the work. Anything that's being done in your body is being done by them. So chromosomes and genes occur in pairs, which we've already discussed. They're called homologous chromosomes. These are the set of chromosomes that we discussed in meiosis, which was a form of sexual reproduction. And the new combinations of genes occur during this sexual reproduction, during meiosis. So fertilization from two parents, so the egg and the sperm, and they each have a set, and they come together, and they make 46 chromosomes in all. So if you'll look at the figure down here below, you'll see that these are the two homologous chromosomes, and these two regions code for the same gene. So say we're talking about eye color. These two areas on these two homologous chromosomes, they code for the eye color gene. So let's say one is blue and one is brown. And you guys know what this is. This is a karyotype, which is what we talked about when geneticists sit around at parties. Remember, I made that joke. But this blow-up is showing that this specific set of chromosomes and blowing it up, and these are the sister chromatids, of course, you guys know that. So homologous chromosomes will have a region on them that both code for the same gene. So let's talk about Gregor Mendel. Who was Gregor Mendel, and why is he important? So his name was Gregor Johann Mendel, and he was an Australian monk born in the Czech Republic in 1822. He was the son of a peasant farmer, which the fact that he was the son of a farmer is going to be key here. He studied theology, which is a religious study, and was an ordained priest in the Order of St. Augustine, which you guys will learn more about in history as you move through your grades. So you guys will see that again. He went to university in Vienna, where he studied botany and learned the scientific method. Now, you guys are familiar with the scientific method by now, I hope, and to you it may be second nature. It's something you've learned in every class since you were in 4th or 5th grade. But at this time when he was coming up and learning, this was something new. This was something that was still being investigated. So he worked with pure lines of peas for 8 years. Pure lines is key here, which means he could trace their lineage back for 8 years. So prior to Mendel, heredity was regarded as blending. They didn't really understand how you could take two plants and they would pollinate each other and wind up with a very unique looking plant. So this was all new to them. They thought it was a blending process, and the offspring were essentially a dilution. They were not pure of the parental characteristics. So you can see how far we've come in science since Mendel's time. So Mendel's peas, and you're going to hear this referred to over and over as Mendel's peas because he's famous. All biology students learn about Mendel's peas. You will see them again if you guys go to college, so don't think this will be your last exposure. But Mendel's peas looked at seven traits or characteristics of pea plants. So as you can see here on this diagram, this shows the traits that he looked at. Rounded or wrinkled, yellow or green, the flowers, were they purple or white, inflated or pinched ripe pods. You can see the ones on the left there. It's full, and you don't see any curvatures, whereas the one to the right there, you see the little stereotypical indentation where the actual peas sit. Green or yellow unripe pods. Axil or terminal flowers. Now you guys don't know what axil or terminal means. Those are botanical terms, but terminal means close to and axil means far away. All right? So is it close to or far away? Flowers. All right? Long or short stems. So that's kind of self-explanatory. I don't feel the need to go into that any further. All right, in 1866, Gregor Mendel published Experiments in Plant Hybridization. He established 3 basic principles of inheritance, which are the building blocks of genetics even today. Even as far as we've come from Mendel's day, these are still pretty foundational, and they have been taken from a theory to law. So we actually call them the laws of inheritance. So he tried to repeat his work in other plants, but it didn't work because the plant then reproduced asexually, which we know creates genetically identical offspring and not unique offspring. So that's an issue. We wanted to see how 2 unique plants would pollinate each other and reproduce sexually. So his work was largely ignored for about 34 years, which is not uncommon for scientists of his day, until 1900, when 3 independent botanists rediscovered Mendel's work. So 34 years later, 3 different botanists repeated this and saw that he was headed somewhere that was right, and they confirmed his work. So Mendelian genetics, we honor Mendel by calling it Mendelian genetics, and it is a special section of genetics. Genetics is very broad, but Mendelian is specific to him. So he was the first biologist to use math to explain his results quantitatively. This is a big deal. Scientists today, we describe our results in numbers and whether or not they are statistically significant, whether or not there is a big enough difference to say this is scientifically a phenomenon. So he was the first to actually track his data, take numbers, and mathematically use that to back his point, to further strengthen his ideals. He predicted the concept of genes and that genes occur in pairs, and that one gene of each pair is present in each of the gametes, right? The sperm and the egg. So he kind of laid the foundation for what we learned in the previous unit. Genetics terms you'll need to know. Guys, I've posted your key terms. They're here again. They're also in the student notes that I hope that you're taking right now while we're having this discussion. So you'll want to copy these down. I'm not going to go read in each one individually, but you will want to make sure that you flashcard these or Quizlet these and study them. Last exam, I think the most of the key terms anybody got were about 9 out of 11. Most of you got about 5 or 6 out of 11. So I will be taking the definitions straight from these pages. So please make sure that you study these. Here's another page of key terms. You should be able to pause this video and copy these down. So I'm not going to spend a lot of time sitting on these slides. Here's another slide of terms. I went ahead and did this for you guys. Please don't make me regret it by not studying them. All right, so let's move on. All right, guys, so here's a buzz question. I want you guys to open up Padlet on your phones, wherever you're sitting, and I want you to type in your name into the title section, and I want you to tell me what the term allele means. Now, you should be able to back up to the key term slides and find this. So this should not, I should get full participation on this one. So go ahead and do that now.